Good morning, Cannabis Public School. Our mission is to ensure that everyone across America has accurate information about the holistic benefits and the potential risks of cannabis. We showcase top-notch cannabis products, important trends, and keen insights, and industry experts. We advocate for increasing acceptance of cannabis therapeutics among conventional healthcare providers to empower individuals to make informed decisions to transform their health. And we embrace the spiritual and recreational value of cannabis. Let's see. We are doing my next project for Cannabis Public School is to get some logos up on our introductory page. So please send me a PNG of your logo as a Cannabis Public School supporter, and I will put that on our homepage. So we start Cannabis Public School with a dad joke, and in honor of our scientists, we have a science dad joke. What brand of underwear do scientists wear? And Anna, if you know this one, do not tell anyone. Calvin Klein, of course. Okay, so if you're like me, you may know that that's a science joke, but you may not know what a Kelvin is. Apparently, it's equal to the fraction 1/27 of the thermodynamic temperature of the triple point of water. I have no idea. But it is the point at which water, ice, and vapor coexist in equilibrium. So good job, Kelvin. Every month, diverse educators share their experience on one of the most groundbreaking advancements in the recent medical history, the potential reintroduction of cannabis into the American pharmacopoeia. At Cannabis Public School, we embrace and spotlight others. To that end, oh, did I just say that? Yeah. So don't know why it's there twice. Today's guest is Dr. Anna Schwabe, aka Dr. Cannabis. Welcome, welcome. We're so excited.
How's it going?
So can our nose detect genetic imposters in cannabis strains?
Keep that bug from
Oh, don't forget to mute. Can our noses detect genetic imposters in cannabis strains? Research from Dr. Cannabis reveals that your sense of smell can detect genetic differences and emphasizes the interplay between genetics and environmental factors. From light and temperature to flower maturity and storage conditions, every element leaves its mark. This is going to be so exciting. Terpene analysis further uncovers variations even within identical genetic fingerprints, highlighting external influences in the final product. This study isn't just about sniffing for fun, it's a roadmap for understanding how your olfactory experience guides your purchases. Let's prepare for a journey where we unravel the sense shaped by the interplay of genetics and environment. Anna, I'm going to turn it over to you.
Cool, thank you. Oh my goodness, thank you so much for having me today. I'm so excited. I haven't talked about this for a while, so this is going to be fun, and I kind of have a new format with my slides. I've redesigned, so if you've seen any of my talks, this is going to be a little different. So I'm going to share my screen.
Yep.
And let's see. Can you see that?
Yeah, it looks beautiful.
Okay, cool. All right, let me. Oh, I don't have any notes, so I don't even need to see that. Okay, cool. So I have it. so I called my talk today Whiffs of Wisdom, Decoding Cannabis Aromatic Variation. And so I just want to give a little quick overview of what I'm going to be talking about today. So I'm going to give you an introduction, which probably will be old hat for most of you who are familiar with cannabis, but I kind of just want to set the stage and make sure everybody's on the same page before I kind of dive into the science. because, you know, I am always surprised when people don't know all the things that I know. So I'm going to start basic, and then we'll dive into the science. And I'll talk about a little bit about genetics and what I found in my PhD research. And then I'll talk about how I selected the genetic samples for this particular study. I'll talk about the sniff panel, the results of the sniff tests, and then talk about chemotypes and then implications for the industry and patients and consumers. And then a little quick conclusion, and then we can do some Q&A.
Great.
All right, so some of the background. So the cannabis plant produces a lot of different chemicals. The ones that generally we're most interested in are the cannabinoids and the terpenes. The cannabinoids are responsible for the majority of the effects, at least we think they are. And the terpenes lend a lot to the flavors and the smells, and possibly also help with creating some effects. And so, you know, I've kind of depicted here the little trichomes. That's obviously enlarged. They're not that big compared to the plant. They are concentrated on the female flowers, and there's a whole slew of them. You know, there's over 100 terpenes. There's over 100 cannabinoids. Those are the things we typically measure, but we also know there's other things such as thiols and alkenes. And, oh wow, that just went weird. Alkaloids and flavonoids and all kinds of different things that we haven't even really dived into. But all of these things together that are present in any cannabis flower are, when you combust the flower, are going to have some sort of effect on each person. And that effect can differ greatly depending on your own personal chemistry and your own personal physiology. That's a whole nother story, a whole nother, you know, deep dive, which I don't do. I don't do people. I do plants.
Sorry.
I'm sorry, I'm gonna mute that situation.
Okay.
Okay.
So I do people, not plants. So the nurses in the audience and the medical folks, that's your job. I'll stay in my lane. And so all of these things together, the terpenes, we know about a bit about terpenes through things like aromatherapy and whatnot. We've got linalool, which is found in lavender. Myrcene is found in myrcene is found in mangoes. We've got caryophyllene, which is found in pepper. Humulene is found in hops. Limonene in lemons. These are also present in cannabis. And then we have the cannabinoids, which we're most familiar with THC and CBD, but there's a whole bunch of others. CBG is the precursor to all of these. And then we've got CBC and CBN, which is a degradation product. And all of these things come together and they work in concert with each other to create effects on the brain and the body and interact with various receptors all throughout your body. And you know, depending on the levels of each of these things, will change the effects. So here I have kind of a little equalizer thing with 6 different terpenes and 6 different cannabinoids, and you can adjust any of these and they're going to change the effects. And these are just 6 of each. And we know there's over 100. And so you change any combination or any level of those, and you're going to create all different kinds of things that can happen. And so it becomes like this really mind-blowing thing to think about, like what ifs, you know, what are the potentials of having these different phytochemicals and in what combinations and what do they do specifically and all of that. And then you've got, you know, the personal physiology that also comes into play. But this is just a very simplistic view of kind of how these things work together. And then, you know, we've got different strains, and, you know, there's probably thousands of them on the market. They have a name. We create them by crossing different other strains. We often measure major cannabinoids and major terpenes. It's become trendy to have like the top 3 terpenes that we look at. So limonene is thought to be energizing. Pinene is thought to help with alertness, and caryophyllene is sort of a calming terpene. But these things are not 1 + 1 + 1. Like if you add these 3 terpenes together, you don't have a calm, alert, energized effect. They work together but it's not a 1+1 situation, right? So kind of how I explain it is that, you cross two things. We know these two things are, you add in external factors. So where was it grown? How was it grown? When was it harvested? And all of these other things. And that is going to shape what is present in the plant when you consume it. So it's an interesting and complex situation that we have. And I also like to throw in that abundance doesn't equal potency. So although THC is generally the most abundant cannabinoid, it's not necessarily the one that is creating the greatest effect. We know now that THCP, for example, is a very potent cannabinoid, and it's thought to be about 30 times more potent than THC. And so some of these minor things, we only ever measure the things that are most abundant. It's like, in a cake, flour, water, eggs, sugar are all going to be prominent in a cake. Is that the thing that makes the cake delicious? Not really. Some of those other things, like, if you added nutmeg or cloves or, a little bit of vanilla or something like that, those are going to shape and really make that cake delicious or disgusting. So, these are things to think about. Just because we measured it doesn't mean that's the thing that's driving the effects. So what causes variation in cannabis? Well, we know if we have different genotypes, obviously that's going to be a big influence on the final plant, the final product. However, there's a lot of external influences that impact that final phenotype, what that plant looks like, tastes like, feels like, and all the rest of it. So that's the phenotype, the things that we can physically see and measure. So the things that shape that are the genotype, obviously, and whether it's a seed, a clone, doesn't matter. And then the environment. And the environment really consists of a lot of different things. So it's the soil, the nutrients, the watering regime, CO2, altitude, sunlight, or artificial light. It's also time at harvest. So, everybody has their own way of determining when something's ripe to harvest, right? And it's not really a scientific method. It's sort of like, oh, when 80% of the trichomes are amber, that's, like, is that really specific? Not really. And some people like to harvest early. Some people like to harvest at exactly the right time for them. Some people like to leave it and get, a completely different product. And I even tell people, hey, harvest some early, keep some around. Like, you're going to end up with 3 different products. And then even post-harvest are going to feed into this final product. So that you would not want to leave your cannabis in a clear glass jar on your windowsill with no lid on, even with a lid on. Like, it's not going to keep well, and it's definitely not going to, all the terpenes are going to be gone. And so storage conditions matter. If you buy cannabis and you find it 5 years from now in a drawer, it's not going to be the same thing as what it was when you purchased it. So all of these things are going to play into your experience upon consumption. So that's the background. So hopefully we're all on the same page. And this last slide, really, this is the crux, the cornerstone of all of my research. And so I had this idea, and I approached my mentor from my master's degree, Mitt McLaughlin, at the University of Northern Colorado. And I said, hey, I have this great idea. Cannabis has just been legalized, and there's very little research on it. And the community is, has all of these problems. The first thing being, when I go to a dispensary, any one of the hundreds that were in Denver at the time, I buy Blue Dream at one, and then I buy Blue Dream at another. They're not the same. What the hell is going on? And I was like, that is really weird, because I always thought that cannabis was propagated, most often through cloning. So they should all be the same, or very, very similar if they were grown from seeds, because they're all siblings, right? And I was like, well, with my background in population genetics, that is a really easy question to answer, because what I need to know is, are they identical? And the answer is yes, or they're not, right? So that's where I started. And my PhD covered, I had lots and lots of questions, and Mitt was very good at reeling me in. You can't answer all the questions. You can't do all the things. Dial it back a little bit. Let's keep it under control. So in the 4 years that that it took me to do all of this work, what I came up with and what I have published, I think all of these since, was the first question, the within strain variation. Are they genetically identical or not? Then we did some work looking at kind of the genetic spectrum of cannabis. So we have wild growing hemp, we have cultivated hemp, we have CBD, and then we have all of the, THC types, drug type that you can get at dispensaries, indica sativa hybrid. So I wanted to look at all of that. And it just so happens that I had a couple of samples from the University of Mississippi, which is the supplier for research grade marijuana through the National Institute on Drug Abuse. And so we did that study. And if you're familiar with it, you know what we found, and it wasn't great. I mean, it was great for us, but not great for the University of Mississippi or the National Institute on Drug Abuse. So then I wanted to know, like, okay, so if we know that within a sample set of Blue Dream, and there's one genetic imposter, does that, one with the weird genetic identity, does it smell different? Like, is it different also phenotypically? Or on the flip side, this would be cool too. Yeah, it's genetically different, but it smells the same. And so maybe that's why it got put in that group to begin with. So this is what I'm going to go over today. And also looking at the actual phytochemicals to see what's driving these differences. And then the THC inflation was recently published. Well, actually, it's been about a year now. And I also wanted to look at some epigenetic changes due to cloning stress. So really what I'm focusing on today are these three. So the within strain variation, the genetic versus aroma variation, consistency difference, however you want to think about it. And then the phytochemical variation, which in theory should go along with, the differences that we smell as a consumer. And so this is just a snapshot. I don't expect you to, you're probably going cross-eyed. This is the kind of genetic tool that I used. Like a lot of people use genomics, which is a lot more complicated than this. This is a pretty easy and cheap genetic method for identifying or looking at relationships between and among individual samples. And they actually use this method for genetic identity of things like fish, olives, apples, tomatoes. Like if you wanted to show a brand new genetic, a brand new variety of apple, you can use this kind of data to show that your new apple is different from other apples. And so really, again, I went around to various dispensaries and just collected samples. So in this sample set, for example, I would have multiple Jack Hares to be able to compare them to each other. And here is some actual data. This is 3 purple Kushes from 3 different dispensaries. And again, what I'm looking for is, are they genetically identical? And whether or not you know anything about what this data shows you, you can just look at it and say, nope, those are not genetically identical, regardless of what this data is showing, right? So game over, done deal. We know the answer. But, in order to publish, of course, we need more stuff to show more validity of that. So here is some of my data. And again, you don't need to know what all of this is. This is published. So if you're interested, you can definitely read it. But we're looking for similarities within a strain. So up here, for example, we have Durban poison, and the 100 just means it's described as 100% sativa. And you'll notice down here that we have grape ape, which is 0% sativa, which means that it's 100% indica as described online, not what it looks like when it grows. Those are 2 different things. So up here, you can see these Durban poisons here, and you can see, the vast majority of them are pretty similar with this blue color, but this individual right here from Denver, it's not the same as the other ones at all. And that's true for 27 of the 37 strains that I looked at. So ChemDog, for example, actually the ChemDog story is really interesting, but I won't go into it. They're all really similar, which was very interesting. Blue Dream, all pretty similar except for this one right here. And so this went on and on with just about everything. Jack Flash, they're pretty much identical, but there's only 2 samples. And I would imagine if I was able to get my hands on more Jack Flashes, and specifically, then we would see there would probably be some genetic variation. And this is just another analysis. The closer together the dots are, the more related they are. And so up here in the very corner, you can see all these red dots. Those are Durban poisons that belong together, but you can see there's a red dot here and a red dot here and a red dot over here. So those are not as similar. And these are all the strains that I got my hands on and looked at. And so really the question here was, can people sniff out a genetic imposter? So although I did all that work on all of the previous work, which is on 122 samples, 30 strains, we didn't have enough to carry that over into a sniff study. We needed 2 grams of each sample to split into 2 different jars to then allow people to sniff them. And so what I had to do is I had to go back out to the dispensaries and drive around and find as many sample. We kind of picked we kind of pre-picked the strains that we knew we could find on the shelves, that we knew many dispensaries would have these on their shelves so that we could get our hands on them and buy them and have enough to do a sniff study. And so this is kind of what we ended up with. Was OG Kush, Blue Dream, Durban Poison, and Mob Boss. We did also have Sour Diesel, but no outliers were found, and we couldn't use that in the sniff study because without an outlier, like we wouldn't be able to do this experiment. So here you can see OG Kush. All of these indicate that they are genetically very similar. That same color represented in this visual representation of the genetic fingerprint, and you can see. So SN means that we have enough sample for the sniff study, and GN just we threw it in because we had enough to also analyze it, so it gave us more robustness to the genetic study to find the outliers. So for the OG Kush, we use these 3 as the ones that were genetically identical, and then we use this one as the genetic outlier. Same thing with Blue Dream. We've got 4 that were genetically identical, and then this one we used as the outlier. This Durban Poison, we used 3 samples, and these were genetically identical, not 1 letter difference in the regions of the genome that we looked at. And so we used those as our control. So in theory, we shouldn't see any difference in the smell of the Durban poisons because they're genetically identical. And then Mob Boss, we use this one as the outlier. Even though it's not very different, it's still different. So then we get to how we did the sniff study. So as I said, we bought enough sample to put 1 gram in. We split it up and had them in different jars because while you're opening and closing all these jars and the participants are sniffing, we want to be able to, in a day or in between, like for each participant, we want to let the cannabis rest and not just have the sample continually open and close because we lose volatile compounds and whatnot. So they were randomly assigned numbers, and the doctor who did this section of the work, Dr. Avery Gilbert, he didn't have any information about any of the strains. The participants were put in a room, no music, no posters, just very boring. We had 40 odor descriptors. We had 55 participants, relatively equal male to female ratio, and check all that apply. What do you smell? And it wasn't done by experts. It was done by random people from the interwebs. But that's fine. Like we all know what apricots smell like or lavender. It wasn't, and it's really not for experts. It's for people, like us who go to a dispensary and we smell something. Okay. So that was a lot of data that I collected, and I had to boil it down to something more manageable for me to look at and get something meaningful out of. So I put those 40 smells, I put them into groups, and the groups were floral, spicy, sweet, earthy, and pungent. And I looked at the genetic identical samples and then the outlier. So the outlier in each one of these graphs is green. Obviously, Durban poison's not in this because they were all identical. And you can see, hopefully, that the outlier for Blue Dream was much more pungent. The outlier for Mob Boss was less pungent, more sweet, and more floral. And the outliers for OG Kush was more pungent, less sweet, more earthy. So yeah, I mean, I can safely say with my data and my groups that the outliers smell different. However, I am not a sensory scientist, and these groups were, I don't know if they're right, if they're wrong. Like, I don't know how you would group them. So I looked online to see if there was something else that was available for cannabis aromas. And this is all I could find was the Western cultured cannabis wheel, terpene wheel. And luckily, most of our odor descriptors fell somewhere on this wheel. So I decided to use their groups of spicy, sweet, sour, and earthy. And as you can see, the outlier, again, in that kind of greenish color, was less sweet and more bitter for Blue Dream. It was more sweet and less bitter for Mob Boss. And for OG Kush, it was just a little bit less bitter than the other ones. And so the answer to the question is, can people sniff out a genetic imposter? Yes, but remember the Durban poison? There is a lot of variation in the smells that were smelled of the 3 Durban poisons that were genetically identical. So we have the mean in green, so you can look at that or you can ignore it. But you can see in any one of these samples, we've got variation from one sample to another: citrus, earthy, woody, lemon. There was a lot of variation. And I know there's not 40 up here. That would make your eyes go crazy. But these were the top smells that were smelled in the 3 Durban poisons. So a lot of variation, even within things that are genetically identical. And so I was like, well, what did the actual terpenes look like? Like, maybe this is something, maybe there's something else going on. Like, let's do some quantification of these things instead of just the subjective sniffing. And so here's the actual terpenes. The Durban poisons, again, were all identical. So you can see any of the variation that's in any of these terpenes is not due to a genetic difference. It's due to something else. And if you remember back, environmental, and we'll get back to that. But, we've got some that are relatively similar. That okay. But like terpinolene, for example, like one of the samples has a huge amount of terpinolene compared to another one. Neurolidol, but I don't know how to say that. For the OG Kush, the green is the outlier. So you can see it's not just the outlier that's different, it's all of them. All of them are different. The outlier for Mob Boss was red. So you can see here, we've got variation from the green to the orange. Blue Dream was relatively consistent among the samples, but still we have variation. So I wanted to, so I was thinking, okay, well, what does Leafly say? Leafly's this huge database that everybody uses. They have the dominant terpenes listed. Keep in mind, Leafly's in California, and these samples are all from Colorado. So Leafly says Durban poison is the dominant terpene for Durban poison is terpinolene. So here's terpinolene. Okay. So in one of the 2 of the samples, dominant terpene, but one of them is not. Myrcene is next. Well, I would say caryophyllene for my samples is up there. And ocimene is right here. I don't know. I don't see that. OG Kush, another one. So they say that myrcene is the dominant terpene in OG Kush. Here's myrcene. But for my samples, caryophyllene was the major terpene. And we've got neurolidol in one of these samples that has, there's a lot of that in there. And then limonene in one of the samples. Mob Boss, the dominant terpene is described for Mob Boss. I would say again that caryophyllene in my samples and neurolidol were the dominant terpenes. Blue Dream, unleafly, dominant. Myrcene, again, I would say caryophyllene, at least for my samples, was the dominant terpene. So we've got some differences. I don't know if it's a Colorado versus California thing, but that's a good possibility. So, again, with the environmental conditions, this seems like it's a very complex problem and makes things really, really difficult. But if you think about it logically, for example, bananas, you can have the same banana. You buy it green, you come back 2 weeks later, it's black. It's the same banana. It's exactly the same individual. Its genotype hasn't changed. But the look, the taste, the smell, the texture of that banana over time will change significantly over the period of 2 weeks, whether you do anything to it or not, even if it's just sitting there. The time of harvest of that banana is going to be, you know, if you harvest it early, if you harvest it late, how you store that banana. Do you just chuck it on your countertop? Do you put it in your freezer or your fridge? Do you put it in with your onions? Like, all of this stuff is going to change your banana, and cannabis is not really any different. And going back to the genetics, even when you start with the same starting material. So let's say I give you a recipe and the ingredients to make cookies. You might follow the recipe to what you think is the tea, but your oven, the kind of cookie sheet you use, the thickness of the cookie sheet, the temperature of the butter, the temperature of the oven, whether or not you farted on it, I don't know. Like, all of these things are going to change the way your cookie comes out, whether, you know, even if you think you absolutely follow the recipe. And unfortunately, some people are just not good at baking. Just like some people are not good at growing cannabis. You know, it's just a thing. So all of these little things really do play a major role in the outcome of your product. And it's not just cannabis, it's so many things. And that's the crux of the show. Nailed it, right? They all get the same ingredients. They all get the same recipe. They even get a picture of what it's supposed to look like. And, you know, the funny thing about it is that so many people just can't do it, right? It's also the basis of the grow off. Everybody gets a clone from the same plant. They also have the same starting material. Someone's going to win that competition. Someone's going to grow that better. Is it from the genetics? No, it's absolutely every single bit of that is going to be the environmental, the external inputs going into that plant to shape it into what it becomes, the yield, the smell, the taste, all of that. And even in my own grow for the company that I work for, all of these are the exact same strain. They're grown from seeds, but every single one of our greenhouses is a little different. And we get a crazy amount of variation in our products. It's all ground down for biomass, so it really doesn't matter. But when we're talking about flower product, it does matter, right? And so just kind of to sum this all up and conclude what I've been saying is, if you start with the same material and then you grow it, you might have some differences because you're going to have different styles, different methods, different nutrients, different light, whatever the case may be. And you are going to get some subtle differences in the chemicals that the plant makes and what eventually will reach your nose. However, it's going to be, you know, it should be relatively similar if you keep everything, all your inputs, if everything on that one side of the equation is the same, you'll end up with something very similar. And we can translate that over into effects. You should get very similar effects. However, if you deviate, if you start out with just a little bit of, you know, a different genetic identity, even if it's just little, these differences are going to compound. So when you're growing it, not only are those environmental influences going to feed into this, but now you've got a little bit of a different genetic identity. So you're going to end up with things that are quite different at the end through no fault of your own, and are quite different for the perceptions of the consumer, for the effects on the consumer, right? So it's really important to, at least for me, I think it's very important that the very first thing that we should be doing when we're talking about a strain or a cultivar or whatever is make sure that we have the thing that we think we have, because it's a problem all throughout the supply chain. At the moment, nobody really has any way to say, okay, I'm ordering Blue Dream seeds. There shouldn't be any such a thing. Blue Dream's too variable, but I'm ordering Blue Dream seeds. First of all, does that supplier know that they actually have Blue Dream? Who knows? Then they send you something. Do you know that that person actually sent you Blue Dream seeds? All the seeds look pretty much the same to most people. You grow it, then you sell it. Does that consumer on the other side of the counter have any protection at all that what they're getting is Blue Dream? And you can't go back to the dispensary and shake it in their face and be like, this isn't the Blue Dream that I'm used to. They're going to be like, oh, sorry, don't know what to tell you. So all of that feeds in. And again, this is a really important equation to think about every time you're thinking about cannabis, because there really are so many variables that create so much variation. And so the conclusions, real quick, your nose knows. You can sniff out imposters. If you are at a dispensary and you're looking for something specific and what they hand you doesn't smell quite right, if you have that opportunity, not everybody has the ability to smell something before they buy it. But if you can, and it smells not quite right, the chances are it's probably not. Go somewhere else or find something else that, seems like it might be the right thing. So I think there should be some consistency requirements, some sort of consumer and grower protections, that there should be genotyping. I think we'll get there eventually. We could implement or at least have standard conditions. If you grow this under these conditions, you will get this, kind of like a recipe. That doesn't mean that you can't deviate from that, but you could say, this is this, but we grew it using our special methods and we have a better product. I mean, that's what they do with the wine industry, right? We know that certain wineries produce better wines because they have their own little special je ne sais quoi that they throw into the growing process or the whatever. And then, of course, consumer education, letting people know, hey, this is a thing that you need to be aware of. It's not the same as buying a Snickers, at 7-Eleven versus, King Supers or whatever. And that the external factors obviously have a huge impact, the growing conditions and then the post-harvest treatments as well. And I just want to say, I know there's probably some people that always cringe when I say strains. Strain in terms of cannabis is a perfectly acceptable word to use. Some people think that it's only reserved for viruses and bacteria. That's completely incorrect. We have strains of rice. We have strains of wheat. And it just means in botany that it's a group of descendants from a common ancestor that share uniform morphological or physiological characters. And that absolutely is applicable to cannabis, right? So I'm going to keep using the word strain. Other people are going to fight me and say they're cultivars. They're not in most cases. And if anybody wants to get into that, I'm happy to debate. And if you want to debate, you can find me here. So my website has all of my information. That's my email. I have multiple emails. You can find me on Instagram or LinkedIn is a good place to find me. And I'm at 33 minutes, so
Wow.
I'll take questions.
Oh my God, we have really good questions in the chat. But first, I wanted to just say, oh my God, mind blown with science, amazing. Anna, yeah, there we are. Cool. Okay, and let's see. I did have something. Everyone loved your slides. Amazing. So helpful for the presentation. I
always worry that it's like too cartoony, not sciencey enough. Like I'm going to get ousted in the science community because, they're too fun. I don't know. Scientists are supposed to be stuffy and boring, but I can't handle that.
We can tell you are a disruptor.
Yeah.
Oh, that's what I wanted to say. I would not want to debate with you. Anyone who could, just, off the top of your head, give a dictionary definition of what a strain is, I would never. Okay, June, go ahead, help us with the chat. And then raise your hand, please. And Christine, I'm sorry, if you want to ask your first question yourself, please unmute. But let's start because there's lots of questions.
Oh, good.
Yeah. Which my Eloise Thiessen, when she was the president of ACNA, when I first joined, and I gave my first presentation, and I said, there were a lot of questions. And she said that usually means that it was a good presentation.
Okay. That probably means that people understood a lot and they're like, ding, 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 like little lights going off. Great. Fantastic.
Okay. Go ahead, June.
Yeah. So the first question, how can the average consumer confirm that they are getting what they say without doing the specific test that you talked about today?
You're not going to like the answer. You can't.
There's no way. Yeah.
There is no way. And honestly, the only thing that I can tell people is if you find something that you like from a particular dispensary, go back and buy as much as you can and store it in your freezer, because you know, you may not ever come across it again. You might have actually gotten the outlier.
Yeah.
And
And
not
that
the one-
that batch may never happen again, right?
and that happens too. I think of cannabis like Lay's potato chips. They come out with those flavors every summer. If you miss the special flavors, like you're not going to get them, like you missed them. So sometimes that happens with cannabis too.
Yeah. I mean, could, I wonder if there's a future where, like, you know, if the smell test works, could they possibly make things where you can sort of like get familiar with the smell and like memorize it, so then when you go to look at the flower, you can sniff out the imposter.
Some people swear that, you know, they can tell, like ChemDog, for example, they can tell if it's ChemDog or not. I would challenge that. There are chemotypes that are pretty well established or are very odd,
Mhm.
to the point that, you know, OG Kush ended up being one of the ones that I was like, what the heck? One of the smells that people often picked up on for OG Kush was cheese, which none of the other samples had cheese as something that people identified. So I think maybe, but you know, the Ganjier program teaches a lot about being able to smell what you should smell, what you
Yeah.
know, and how to smell mold, what you shouldn't smell. And I mean,
What program is this?
huh?
What program is this?
It's called the Ganjier program. Uh,
Okay.
like sommelier, but like for
Yeah,
cannabis.
sure.
Uh,
I was
it's
gonna say that as well. I know for sommeliers and stuff, you can't really be like a master sommelier unless you have like specific physiologically certain traits as well, right? Isn't that
yeah.
correct?
Yeah. So the Ganjier program, it's a set of courses that you do at home, but then you have to go in person. I think it's for 1 or 2 weeks. And
Okay.
you have your hands on the cannabis. You are taught to look at it, smell at it, touch it, taste it. You have to smoke it, which is probably why I haven't gone through it, because I don't think I'm THC and I don't really mix very well. And I don't
Okay.
think I could smoke all the cannabis that's required to get through the program. But I am friends with several of the instructors and Kevin Jodry, who's the founder. And from everybody I know who has been through the program, apparently it's incredible.
Okay. And it's
Highly
in Colorado?
is what?
I'm sorry, it's
Oh,
in Colorado?
we'll definitely talk about that later. Let's get on
Yeah.
to the
Oh,
questions,
sorry.
because I have a quick question.
Okay.
And it kind of follows this. I've been intrigued with this snippet of information I read somewhere, and of course didn't save it, but it's saved in my brain. And basically, it's about the external, and you're a geneticist, so maybe you can help me with this. The external environment that nurtured you as you grew up shapes what you smell. So therefore, someone who grew up in the desert just does not have the olfactory receptors, the same olfactory receptors as somebody who grew up in the forest. So A, is that true? And B, would smelling cannabis for a living create new olfactory receptors?
I mean, isn't that just a experience, or isn't that just a
Oh,
memory that you create from, you know, having smelled a fig before or having smelled vanilla before?
Kim, I love you, but can we let the speaker answer? Thank you.
So, that makes a lot of sense that if you didn't grow up around a certain smell that then you wouldn't have, you know, you wouldn't be familiar with that smell. So, potentially, but I don't think, I mean, I think once you've been exposed to a smell, you're not going to smell pizza that you didn't grow and be like, I can't smell anything. Like you can smell it for sure. You just need to know what that experience is and then be able to put a label on it for yourself. Like we have no idea. Like we have no idea if you and I, when we smell the same thing, both actually smell the same thing, but our brain has interpreted what that smell is. And then we put a label on it. That is pineapple. Doesn't matter what it smells like to you. Doesn't matter what it smells like to me. We both smelled it and we both know that smell is pineapple, right? So the other thing is that we had a huge variation of people in this study. And we also measured the terpenes. So whether or not the people differences matter should have been washed out when we did the analytical stuff. Potentially, unless there's other things going on with the cannabis that we didn't measure.
All right. All right, I'll have to go over the recording for the answer because I was so distracted. Okay, Megan, go ahead and ask your question.
Hi, Anna, you're amazing. I love you. Question. What do you have to say about that paper that came out last year on like that it's actually not the terpenes that's creating the smell, it's like the other aromatic compounds. I think scatol was one of them. Do you have any insight on like that new information that came out?
Yeah, so it's actually not completely new information. There was a paper, and it's up there somewhere, that came out a few years ago that one of their conclusions was that the terpenes really only were responsible for about 50% of the variation in the cannabis that they like. It didn't explain all of the variation, didn't explain all of the information that they were gathering. There was more stuff in there that was lending to what they were measuring. And so when that paper came out, and ever since I've read that, I'm like, dude, that totally makes sense. Like, a lot of terpenes are volatile, and when you smell it, what you're smelling is those terpenes leaving. So they're not in the cannabis anymore. Also, we light it on fire. Like, what is on a COA and what we measure is not at all what we're experiencing. And I think it absolutely makes sense that some of these things that are found in very, very minute quantities, some of those styles are parts per billion. We can pick up one molecule in a part per billion sort of situation. Like, yeah, of course, they're lending a lot to these experiences, and we're never going to test. I mean, maybe one day we'll test for them, but we're not even looking for them yet because they're not even there really, you know? So it made a lot of sense, and I do applaud those researchers for that paper because it made a lot of light bulbs go on for a lot of people. They're like, oh, holy shit, that's crazy.
Awesome.
June,
Thank you.
more questions in the chat?
Yeah. The next question was, were the batch dates all the same for the smell test?
The batch date? They were all purchased. Oh, so, okay, so they were all purchased in the span of a week from different dispensaries across the Front Range. Like, the purpose was to get different things. We didn't want to go to one dispensary and only test, like, we wanted differences. So no, the batch dates probably weren't the same. Yeah.
I would interject here, doesn't a lot of it matter whether you purchased sun-grown versus indoor? Because all plants, well, part of their terpene profile is also their defense system, correct? When they're growing outside? Yes?
Well, for cannabis, that is debatable because we've been cultivating and shaping what the cannabis that we have today
Right.
for thousands of years. Cannabis has been coddled and cultivated and looked after and chosen by us. So all the terpenes that are present in cannabis are not for insect defenses. They're for us. We've chosen them based on our preferences. In the regular plant world, yes, terpenes are a part of the way that plants communicate with each other and also fend off some predators. But I would argue maybe not for cannabis. Especially dispensary cannabis. Those are all chosen by us.
Right. Your next question would it be useful, or what difference would it make if all seeds were purchased from a certified seed breeder, meaning the seeds are certified to be distinct, uniform, and stable?
Yeah, so that's what normally happens with plants and cultivars. Like, you get certified, you show that it's unique, distinct, all that stuff. And this is inbred lines. So you take two inbred, cultivars that are very homozygous, very little genetic diversity within these parents. You cross them, and then you do some more work. You breed that new thing, and then you register it. None of that happens in cannabis. So if we think about dog breeding, you take a dog breed, which is a Dalmatian, let's say. A Dalmatian breeds with a Dalmatian, you get more Dalmatians, right? There's no genetic diversity within Dalmatians. You breed that with a Dachshund. You breed a Dachshund with a Dachshund, you get more Dachshunds. There is a little variation, coat color, coat, whether it's short hair, long hair, wire hair, a little bits of size differences. But for the most part, it's a Dachshund is a Dachshund. You breed that with a Dalmatian, you are going to get puppies, but they're going to have differences, right? They're not all going to look exactly like a 50-50 split between a Dalmatian and a Dachshund. You're going to get variation. Then you take your pick of the litter, you breed that back to one of the parents. And then you keep having litters and you keep picking out the ones that you want. And that's how you kind of create a new cultivar. We don't do that with cannabis. We take polyhybrids, which just means a strain that we like. We cross it with another strain that we like. We pheno-hunt the offspring. That's a new strain. So if we took the steps and did the work, which takes a lot of resources, a lot of money, a lot of time, potentially, unless you're using genetic, tools. Yes, we could get there where we have certified types. We have seedsmen who can certify and distribute those seeds. Then we would be at that point where we're at with like tomatoes. If you buy a packet of beefsteak tomatoes and you grow them at home, you're going to get beefsteak tomatoes. And we know that. We're not there with cannabis yet.
Okay. Next question. Does coffee also work between the scents, like, how you do it with perfume? I guess not a palate cleanser, but
Yeah.
Yeah.
Yeah, I know what you're saying. I don't know. I didn't do the sensory portion of the study. But I can tell you the reason that we only did 15 samples is because your nose can only handle so much before it kind of peters out and everything kind of starts to smell the same. But I don't know if there was a nose cleanser in between each sniff or how that's conducted. But that's Avery's wheelhouse, and he conducted the study in the way that those kinds of studies are conducted. So I'm assuming that he did it the right way. But I don't know if coffee was used or, I don't know the answer to that. That's a good question, though. I should probably know the answer to that.
Yeah, I was just gonna say, Kelly, when you guys were working in Gangier, do you use anything between?
Yeah, they actually did give us some coffee grounds, little thing of that to refresh our scent a little bit. So, and I feel like they also kind of either exercise or kind of using to just smell your skin or something like that, or just just wanting something just neutral, I guess, to kind of refresh your nose, um, to practice for smelling different cannabis samples. So.
Mm-hmm.
Yes. So make you smell something different for a second and then go back to smelling.
Yeah.
Yeah, and coffee was actually one of the scents that was on the list that they could choose. So I don't know if coffee would be a good choice or a bad choice in that case. But yeah, I need to ask Avery now. Hold on, I gotta no, I'm just kidding.
Yeah. Maybe it's maybe just normal coffee ground, nothing crazy or anything, probably, but
It's funny that we're still talking about using coffee in between when people used to bury it in coffee, hoping that it wouldn't get picked up, back in the day when it was being shipped everywhere, it was buried in coffee grounds. Or lots of drugs were. I just kind of chuckle at that. Next question, is there a data center where you can find the genetic mapping that each strain is supposed to be, or are they just being compared to one another?
I mean, yes. So they are being compared to one another. There is not what it's supposed to be. So cannabis has been so messed with by humans for so long that there really isn't anything even out in the wild growing that would be untouched or unpollinated or unadulterated from cultivated cannabis. So we don't really have anything to compare it to like we do with other things. So wild strawberries, for example, you can go hiking in the mountains and you can find them, right? So we can compare strawberries to those wild growing ancestral type strawberries to figure out what it should be. We don't have that with cannabis. So that makes it, that another layer on top of the already complex puzzle. But there are a couple of databases that have been, gathering genetic information. So the first one was Phylos, and they did, something very similar to what I did, but they don't do that anymore, and they've taken down the Galaxy. Then there was the Cannapedia from Medicinal Genomics. It's really hard to navigate and very difficult to read that database. And honestly, you as just a regular person, you wouldn't get anything out of it that would be useful for you as a consumer to tell whether or not you have the right thing. Potentially as a grower or cultivator, you could send in something, but it's very expensive. Like to get your stuff tested, it used to be like $5,000. I think it's come down to about $2,000, but to test everything that you're going to be growing, like that's incredibly expensive. There are some other ones that have come online. MyFlora DNA is doing genetic work and databasing. They also do testing. And then LeafWorks is another company doing great things with genetic testing. They also have a herbarium, if anybody knows what that is. It's like a library for plants. They press them, dry them, press them, and you can go and you can take those plants, look at them and then compare them. So if, honey says, I'm growing this, we can go, oh, does it look like the one that's in the database and the one that's in the herbarium, and kind of get the information that way. So yes, there are some people doing it, but there is no central, like the, what is it? The Plant Variety Protection Office, where they keep all that information kind of in a central location. There isn't anything like that really for cannabis. It's just people kind of getting their things up and doing their best to compile the information.
Right. So yeah, before we end the hour, a couple of quick, I do like that Brianna asked this because you touched on this earlier. Do you think there's a difference between strain and cultivar?
Absolutely.
You want to elaborate?
Yes, I do.
Quickly, just quickly.
So cultivar, like dog breeds, like you can take two dogs and you can breed them and you can have puppies. They all exist, and you give them all names. It's Charlie and Samuel and Sissy and Spot, right? They are all things, they all exist, they all have names. To me, that's the equivalent of a strain. However, if you've got these two dogs, you breed them, you have puppies, and Charlie is just amazing, and you want to make Charlie into a new breed, you have to do more work. You have to backbreed and then breed and then backbreed and breed and backbreed and keep picking out the dogs that are the closest to Charlie, so that eventually when you breed two Charlies together, you only get Charlies. To me, that's the equivalent of a cultivar, where you have something that is very stable, uniform, different than everything else. And that to me is the definition, and is the definition of a cultivar. And that is not what we have in cannabis. You'll see things like F1 generation. That just means that two things are bred together. Well, no, it doesn't really, but it gets more complicated than that. Most F1s in cannabis aren't true F1s. They're not uniform. They're not stable. You breed them together, you're going to get a whole mishmash of stuff. Anyway, to me, no. And that's why I use the term strain, because they're not equivalent to cultivars. And cultivar is an actual taxonomic rank. Like there's species, varieties, cultivars, they get elevated and recognized taxonomically in the books. That means that, I can't even think of something like Blue Dazzle Sparkle would be recognized in the books as like a new thing. And no, no, it's just a polyhybrid cross. It's a strain.
Right. I have met very few growers, and these are usually people who have been growing for a very long time, that have even talked about back breeding, as you mentioned earlier. And because a lot of people aren't doing that. So that's a good way to kind of differentiate. And I think I've got one more question. Hold on. Oh, do you think the processing of cannabis into concentrates, even the least processed, like a hash, resin, rosin, keep the same effects and the terpene profile as the source flower?
I don't have any clue. I don't do testing on processed stuff.
Right.
I'm not a chemist. So I don't
Right.
know. I don't know. Somebody else can do that work. Sorry. I think
That's
honey does.
okay. Go
I
ahead.
think
I got a question. I didn't actually put it on the chat, but hey, Anna, it's so much fun to see you, darling. And I love your science. Love, love, love your science. And you just cracked me up to no end when you had to tell Dr. Sulak the other day that you were a geneticist. Remember that? Oh,
yeah.
my Lord. After you've just gone through this big, long diatribe anyway, can you help me get over my sort of COVID attitude about backflush from smelling cannabis that everybody else smelled that day?
No. I mean,
Right.
if that bothers you, then yeah, I don't know. I don't know what to tell you, but I
Is
mean,
it not a problem?
it's definitely not ideal. We don't want to be sticking our faces in a jar that 17, however many other people have had their faces in. People are not generally very clean or sanitary. I mean, no ill will or no
No.
judgment. But in general, people are not that clean. So no, I wouldn't want to do that either. Um, yeah, I know in Colorado they're supposed to switch things out, but still it's the same jar. It's like, yeah, it's pretty nasty. No, I'm with you, girl.
Okay. Thanks. Appreciate you. Love
Yeah,
you.
as someone who studies infectious disease pretty closely in the hospital, I worked in an infectious disease setting. I am concerned about that, but we don't, otherwise we'd all be getting like crazy sick all the time, so there's some barrier of defense, but I don't know if we are like
I
lowering that threshold or what.
So I
You
would
know.
love to get to a point where you can recreate the smell of like a certain type of cannabis and put it in a spray jar and do it like perfume, like, like spray, spray, spray. Oh, I like the smell of that. Or spray, spray, spray. Oh my God, that smells like teeth. I don't like that, and then you're not doing the germy thing. So I would love it if we could get to that point. I mean, I think that's uh, a really cool,
Brilliant idea.
especially with all the terpene companies. And we know it's not just terpenes, but if we could get scientists to kind of recreate flavors, even if it's mostly correct, like it would give people a very good idea of what they're taking home.
Well, we know they're doing that in food additive.
I mean,
they're creating cherry pie. Cherry pie
Yeah.
from McDonald's is not from cherries; it's from
scratch and sniff stickers, right?
Yeah.
I mean,
yeah.
it's not a new concept.
Oh my God, is this your elevator pitch for your $1 million seed money or whatever?
Yeah, well, I love the idea and I think it would be very helpful. However, I'm not sure that we're there yet in recreating the smells. I have purchased and worked with a couple of different terpene companies, but the fact remains, like if we came up with peanut butter breath, for example, not every peanut butter breath smells the same across the country from grow to grow. So you would really have to send in, and get and that could get very expensive again. This is what this batch smells like. The next batch could be different, and if you want to kind of keep on top of that, I don't think you could just do, oh, this is peanut butter breath and then send it far and wide, because I don't think it would match up that well.
Honestly, I feel like we could talk all day, but I do want to respect everyone's hour. Anna, I put your digits in the chat. Are you open to receiving questions? Because there was a lot more that we haven't got to.
Yeah, sure. You can either, I don't know if you want to compile them and just send me one thing, or if you want people, I mean, like I said, I'm pretty easy to find on LinkedIn. So, and I'm always on my freaking phone, always on, the socials. So if somebody sends me a question on LinkedIn, I just can't help but answer people when they text me. So
nice.
Yeah, thanks, Anna. You're always fun. I've caught you on a couple of other webinars before. I like how you present things.
thank you.
You're welcome.
Do you send out slides? Or are they proprietary?
No, you can share them. There's nothing in there that like isn't published or, yeah, no, that's fine. And there, so although it is PowerPoint, I do it a different way. So you can't mess with the slides. The whole thing is just one picture. So
Have at it.
there you go.
Amazing. You're getting so many kudos in the chat here.
Thank you. I should read the chats.
So while you're reading the chat, we always end Cannabis Public School with a quote. This one comes from Henrik Skolomakowski. Let's see what the drop down says. He is, don't know who he is. It's a mashup of two quotes. The first act of awe. It says when man, but I'm trying to think of what to say instead. When humans were struck with the beauty or wonder of nature was the first spiritual experience. We can use science to become spiritual. Many have. If I had the influence with the good fairy, I should ask her gift to each child in the world to be a sense of wonder so indestructible that it could last throughout life. That comes from the author Rachel Carson. Okay, get out there, everyone, get out there and have an indestructible sense of wonder.
Thank you so much.
Bye, guys. Have a great day. This was amazing.
Thank you so much. Bye.
All right, everyone. Bye, Michelle.